Hi students, today we have to talk about another chapter, namely the animal kingdom. Now we have millions of individuals or the animals in this world. To study and describe the different animals, there is a need for classification. Not only that, but also we need classification for assigning systematics for the newly arrived animals. Now, what is the basis for classification? So we need certain basic principles to classify the individuals. What are the basis? Number one, that is the arrangement of cells. How are the cells arranged? Either loosely arranged or compactly arranged to form tissues or organs. Number two, body symmetry. The nature of the symmetry, either radial or we have asymmetrical or bilaterally symmetrical. Number three, the nature of coelom. Coelom is a body cavity lying between the gut lining wall and the body wall lining. And number four, the patterns of different systems in the body, the physiological systems, the digestive, respiratory, circulatory, etc. These are all the different, actually the basis, or we have the basic principles in a crude way for classification of organisms. What are the different levels of organization? We have either cellular level of organization or tissue level of organization or organ level of organization or organ system level of organization. All these organizations are with reference to multicellular animals, not unicellular animals. In some animal groups, say an example of sponges, the cells are loosely arranged. And the cells are in the form of aggregates without formation of tissues or organs. That level is called cellular organization level. That is in the case of sponges. Though the cells are arranged, grouped, and every cell has its own function. That means there is a division of labor among the cells, though there is no formation of tissues or organs. The second level, if we are taking the coelenterates like Hydra, the cells are organized to form tissues. So there is a level, what's called tissue level of organization in the case of coelenterates. You see that one in coelenterates, there is a tissue level of organization. So, and they perform definite function, as in the case of coelenterates like Hydra. The third level, the tissues are organized to form what is called organ level. That is seen in the case of, say an example of the flatworms, the platyhelminthes animals, and also in some other what is called higher group of animals. So anyway, a still higher level of organization is the organ level, and that is being exhibited or shown by what we have platyhelminthes and other higher phyla, where tissues are organized to form organs. A still further higher level in the case of annelids, arthropods and mollusks and echinoderms, along with the non-chordate, hemichordates and chordates. We have the organs are organized to form organ system level of organization. We have definite organ systems like digestive system, respiratory system, circulatory system, etc. Though the animals have, actually different animals have organ system level of organization, they show various patterns of complexities. The complexity is increasing from lower level to higher level. You see that one? Different groups of animals exhibit various patterns of organization or complexities, we can see. Sorry for the inconvenience. So we have just normally, we have the different organ systems. Though we have the organ systems in different groups of animals, normally they exhibit various patterns of what is called complexities, from simple to complex complexities. For example, in the case of flatworms, like what we have the liver flu belonging to the Platyhelminthes phylum, they have only single opening, just which is acting as both mouth and anus. So that the digestive system is not fully developed, it is called incomplete digestive system. It is incomplete. But you can have complete digestive system with both mouth and anus. In the case of higher animal groups, say an example we have, that is annelids, arthropods, mollusks, echinoderms, chordates, etc. They have complete digestive system. So another what we have complexities regarding the circulatory system. A simple circulatory system is seen in some groups of animals. That is called open type of circulatory system. Normally here there are no blood vessels. The blood that is pumped by the heart, in some cases heart is absent, and the cells, actually it is bathing the cells and tissues. Okay, so the blood is pumped by the heart, the cells and the tissues are being bathed in that blood. So no blood vessels. In contrast to this one, complexity increases with increase of what is called higher animals. And in the case of this one, we have closed circulatory system. That is, the blood is passing through the blood vessels of varying diameters, either by the capillaries or by the arteries or by the veins. They are being supplied, the blood being supplied to different parts of the body through the blood vessels in the form of various diameters of arteries, veins, and then capillaries, etc. So anyway, this is what we have, the levels of organization. Even in the organ system level of organization, we have differences in complexities from simple to complex. Now the next one, symmetry. The mode of arrangement of different organs and organ systems in different parts of the body, regular arrangement showing left and right side, and that is called symmetry. There are different types of symmetry seen in animals. One is asymmetry, then radial symmetry, and the third one bilateral symmetry. In asymmetry, you see that one, any plane that passes through the center or central axis does not divide, does not divide them into equal halves. So any plane that passes through the center or central axis does not divide them into equal halves. Such a symmetry is called asymmetry. Asymmetrical animals are the sponges. Or we have the poriferans. The phylum Porifera, which includes sponges, normally exhibit asymmetry. So in some cases, actually, when any plane that passes through the central axis divides, the entire organism into equal halves is called radial symmetry. Okay, does not divide in the case of asymmetry, but divide into equal halves. When any plane passing through the central axis or center of the body divides the organism into two equal halves, and that is called radial symmetry, as seen in the case of three phyla. It's an example of Ctenophora, Coelenterata. You see here, Ctenophora, Coelenterata, Ctenophora, and echinoderms. Coelenterata, for example, Hydra. Then Ctenophora, for example, you see that one, Chlorobrachia, or we have sea combs, comb jellies, walnuts, sea walnuts, or we have what's called echinoderms like starfishes. They exhibit radial symmetry, these three groups of phyla. Now, in higher animals, you see that one, animals like, for example, higher animals like annelids, arthropods, mollusks, and also we have the non-chordates and non-chordates like hemichordates and chordates. The body is divided into two equal halves, right and left. When a plane of symmetry is passing through the central axis, you see that one, this is a central axis, which divides the body into identical left and right halves, Now this is the radial symmetry. You can see that one, the body is divided in two. There is two equal halves in any plane which passes through the central axis, as seen in the case of Coelenterate or Hydra. Here the body is divided into two equal halves, right and left, when a plane of symmetry passes through the center or central axis. So it's seen in the case of annelids. Just even the case of platyhelminthes, annelids, arthropods, mollusk, and also we have hemichordates and chordates, such a type of symmetry, bilateral symmetry. Normally, in the case of animal, there is another method of classification based on the nature of what is called body wall during embryonic development. In some organisms, during embryonic development, what will happen? The body wall consists of two germinal layers, namely outer ectoderm and inner endoderm. Such animals having two body wall layers are called diploblastic animals. For example, Coelenterata, Ctenophora. Even in the case of some of there is what we call sponges also, but they are not coming under what is called the radial symmetry, but they have diploblastic body wall. Now, in the case of such animals, diploblastic animal, there is no middle layer called mesoderm, but there is a non-cellular layer, which is jelly-like. That is present between the ectoderm and endoderm. This is not differentiated into cells, and that layer is called what is known as mesoglia. So ectoderm, endoderm in between, non-differentiated or undifferentiated mesoglia, jelly-like. As seen in the case of coelentrates, ctenophores, coelentrates and ctenophores we have. Now, there is a third layer is present in the case of higher animal groups, that is between the ectoderm and endoderm, that is called mesoderm. So, in between the ectoderm and endoderm, there is a middle layer, what is called mesoderm in the case of higher group of organisms. And such animals are called triploblastic animals, having three-layered body wall during embryonic development. For example, in the case of, so we have ectoderm, endoderm and mesoderm. For example, in the case of platyhelminthes, asculminthes like roundworms, annelids, arthropods and mollusk. Okay, then chordates, non-chordates. We have triploblastic condition including the echinoderms. Though echinoderms, the starfishes have radial symmetry, they have triploblastic condition regarding the body wall. Now we see that one, just actually the body wall lays, you can see outer ectoderm, inner endoderm. Now we see that one. The first one, diploblastic. So outer ectoderm, inner endoderm, in between which non-cellular jelly-like mesoglia. Now in the case of triploblastic, outer ectoderm, that is inner endoderm, in between a cellular mesoderm. That's why it's called triploblastic condition. That's the main difference between these two. No mesodermal layer, undifferentiated jelly-like mesoglia, and here there is a well-differentiated mesoderm with the cells. Now the next category, what's the basis? Coelom. Coelom is another basis for classification. So between the outer what is called lining of the body wall, inner lining of gut wall, just we have the lining, simply we can say the lining of gut wall and the lining of body wall. Between these two there is a space. That space is called a coelom. If that space or body cavity is lined with coelomic epithelium derived from mesoderm, or simply we can say if that body cavity is lined with mesoderm, that is called true coelom, and the animals are called coelomates. For example, annelids, arthropods, mollusk, echinoderms, then chordates, hemichordates, all have a true coelom present between the outer body wall and then inner, that is gut wall. Now this is a true coelom, outer body wall lined with what is called mesoderm, the red one, and here the gut wall lined with what is called the mesoderm again, and this is actually the mesoderm. This space is called true coelom. Now in the case of animals, so true coelomates are annelids, arthropods, mollusks, echinoderms, chordates, and also non-chordates like, that is hemichordates. Now in some animals, now actually what is happening, there is no true coelom. That means there is no mesoderm lining the body cavity. Now actually the coelom is found in the form of pouches. Now these are all coelomic pouches, not found continuously, coelomic pouches. And here there is no mesoderm. Such a coelom that is without mesoderm but present is called false coelom or pseudocoelom, and the animals are called pseudocoelomates. So an example of roundworm, you can see in the case of roundworm, which comes under nematode, that is phylum Ascomynthus. So here you see that one, the coelom is found as patches, no mesodermal layer as here the red represented, and that one is absent. Now the last one is some animal groups, an example of the flatworms or platyhelminthes. Even the body cavity is absent. We have only outer ectoderm and inner endoderm, in between which there is no space. That space is filled with what's called parenchymatous cells. The space is filled with parenchymatous cells. That's why now the coelom is completely absent. Now this is the parenchymatous cells, outer ectoderm, inner endoderm. There is no coelom. The coelom is filled with parenchymatous cells. And that is what is called pseudocoelom. Sorry, I made a mistake. That is acoelom. No coelom is absent. As seen in the case of flatworms, the group, the phylum Platyhelminthes, like liver fluke, the tapeworm, etc. That's about the classification based on what is called coelom. Again, based on the segmentation. In some animals, the body is divided linearly into many segments, and each segment has a representative part of all the systems, or we can say a serial repetition of what is called at least some organ, serial repetition. Organs are present a part in each segment. So that is called serial repetition of what is called, that is at least some organs. So linear division of the body into many segments, and all the segments are alike, having a representative part of all the systems. A pinch of digestive, a pinch of circulatory, a pinch of reproductive, a pinch of excretory, like that in each, that is segment. And such one is called the phenomenon metamerism. What is metamerism? The linear division of the body into many segments, and each segment has a representative part of all the systems, or we can say that is with a serial repetition of at least some organs because of the linear division of the body. Here we have, and that is called metamerism. Animals with segments are called segmented bodied animals, and others are non-segmented. Now if you are taking chordates, there is a presence of dorsally located, a rigid notochord, a rod-like structure which then replaced by vertebral column in higher vertebrates. Now this notochord is a rod-like structure filled with the vacuolated cells derived from mesoderm present dorsally to what is called the digestive system. So there is a development of notochord in some animals. So the animals with the notochord are called actually chordates with the notochord. And other animals which do not possess what we call the notochord are called non-chordates. For example, hemichordates like belanoglasses. Okay, so that's about what's called notochord. Notochord is absent in non-chordates and present in the case of chordates. Now we have to go for the classification of animals, the second one, and then I want to just conclude this one, the major principles or the basic principles used for the crude classification. Now we have to classify the organisms based on certain principles, the next one. And here we have to represent the flow chart of whatever things we have studied so far. See that one, I mentioned already, we have the animals, multicellular animals. If you are taking the kingdom Animalia, see, if you are taking the levels of organization, we have two levels. One is called cellular level, another one tissue level of organization. You see that one under the animal kingdom. If you are taking cellular level of organization, regarding the nature of symmetry, mostly they do not have any symmetry, so asymmetrical. Even the coelom is absent, no coelom in the case of what is called cellular level of organization. So we can say body cavity is absent, so acoelomate. The best example we have porifera, the animals are sponges. The second category, what we call the tissue level or organ system level. So under this one, based on the symmetry, we have two types. One radial, another one bilateral symmetry. Now if you are taking the radially symmetrical animals, the group is called radiata, also called by another name what's called diploblastica, because they have two-layered body wall. That's why they call it diploblastica, and they do not have any coelom. So you see that one, radial symmetry, acoelomate condition, diploblastic condition, seen in the case of Slendata, otherwise called Medaria, and another one Ctenophora. If you are taking the bilateral symmetry, The animals are bilaterally symmetrical and based on the body wall layers, they are triploblastic, having three-layered body wall, ectoderm, endoderm and mesoderm. Regarding the coelom, the bilaterally symmetrical animals are classified into acoelomate animals without coelom, as seen in the case of platyhelminthes animals like flatworms. In the next category of bilaterally symmetrical animals, what we have with the false coelom or pseudocoelom, they are called pseudocoelomates and actually triploblastics. All these three are triploblastics, though the coelom is different. All these three are what we have triploblastic animals. Now with the false coelom, pseudocoelomates, example Ascalminthes. Okay, and what about the nature of the coelom? Normally during development there is one stage what is called blastula, which encloses a cavity what is called blastocoel. The blastocoel then transformed into orchenteuron, that is normally during the next stage what is called gastrula. So blastula with the blastocoel. Now normally the blastocoel is replaced by orchenteuron in the case of next stage gastrula. But in the case of pseudocoelomate animals, the blastocoel is not transformed into what is called orchenteuron. So it remains as such. That's why it's called what is known as the remnant of the blastocoel. The pseudocoelom is nothing but the remains of the blastocoel. It's not being transformed, it remains as such even in the adult also. That is called what is known as pseudocoelom, as seen in the case of Ascalminthes animals. So we can see the pseudocoel is nothing but the remains or the remnant, R-E-M-N-A-N-T, remnant of the blastocoel, the embryonic blastocoel. Normally we can say the primary body cavity is called blastocoel. The primary body cavity is called blastocoel, as seen in the case of blastula. The secondary body cavity is otherwise called as coelom. The secondary body cavity is otherwise called as coelom. Now the third category, what we have actually animals with the two coelom, that is animal called coelomate animal. Here also we have two different groups of animals based on the nature of formation of coelom. One actually enterocoelom, another one eucoelom, simply we can say, or schizocoelom. Based on the nature of coelom, the two coelomates are again classified in the schizocoelomate. Another one enterocoelomate. The schizocoelomate animal, the coelom is developed by the splitting of mesoderm, schizo, S-C-H-I-Z-O, that is called the splitting. The coelom is developed by the splitting of mesoderm. For example, the case of annelids, arthropods, mollusk. The coelom is called schizocoelom, formed by the splitting of mesoderm, but a true coelom. But in the case of echinoderms and hemichordates along with chordates, the coelom is called what is called enterocoelom. That means the coelom is developed from the orchenteuron. That is a cavity of gastrula is called orchenteuron, from which only the coelom develops, hence called enterocoelom. The orchenteuron only opens the outside through blastopore. So blastopore is the opening of orchenteuron found in gastrula, not in blastula. Though it is called blastopore, found only in gastrula, the opening of orchenteuron is called blastopore. So, based on the nature of development or the fate of blastopore, the animals are classified into protostomes, P-R-O-T-O-S-O-M-E-S, protostomes. Another one, deuterostomes, D-A-U-T-E-R-O, deuterostomes. Protostomes means here the blastopore develops into moth. Moth is formed first, as in the case of annelids, arthropods, mollusk. In the case of echinoderms, then hemichordates and chordata, the blastopore develops into anus. Mouth is formed at a later stage of development. They are called deuterostomes, D-A-U-T-E-R-O, D-A-U-T-E-R-O-S-T-O-M-E-S, stoma, mouth. So, in protostomes, mouth is formed from the blastopore, and in deuterostomes, mouth is formed at a later stage of development and not from the blastopore. The blastopore develops into anus. These are the fundamental basic methods of classification. We have the classification of animals in the next what we have video. Okay, thank you.